Hello friends, in the last video we discussed about introduction of translation in prokaryotes. Here we are going to discuss about steps of protein synthesis in brief. Then we will discuss about events involved in initiation in detail. We will see the explanation of initiation by diagram, and then we will discuss some facts and terms related to initiation. So stay tuned. There are three steps of protein synthesis: initiation, elongation and termination. Let us see the events or steps involved in initiation in detail with the help of this flowchart. Initiation factor 1 and 3 binds to 30S ribosomal subunit. Then it binds to messenger RNA via Shine-Dalgarno sequence and move 3' along the messenger RNA. That is, it moves 5' to 3' direction along the messenger RNA until it locates the initiation codon AUG. Then initiated transfer RNA binds to the initiation codon. This initiated transfer RNA is charged with N-formyl methionine and it is complex with IF2 and GTP. This IF2 facilitates the binding of initiator transfer RNA to the initiation codon. Then initiation factor 1 and 3 are released. Now 50S ribosomal subunit binds with the release of IF2 and hydrolysis of GTP. And this binding of 50S ribosomal subunit to the 30S ribosomal subunit form 70S initiation complex. Then it is ready for next step of protein synthesis, that is elongation. Now let us understand this initiation step with the help of this diagram. Initiation factors bind to 30S ribosomal subunit. First binding of initiation factor 1 and 3 takes place. Then this 30S ribosome binds to the messenger RNA via Shine-Dalgarno sequence and it moves along messenger RNA in 5' to 3' direction till it locates the initiation codon, that is AUG. So this is the Shine-Dalgarno sequence and this is AUG. Then binding of initiated transfer RNA takes place, which is charged with N-formyl methionine and is complex with initiation factor 2 and GTP. This initiation factor 2 facilitates the binding of initiator transfer RNA to the initiation codon, that is AUG. Then initiation factors 1 and 3 are released. This is known as 30S initiation complex. After that, initiation factor 2 is released and GTP is hydrolyzed. With this, 50S ribosomal subunit comes and binds with this 30S initiation complex. Now it is known as 70S initiation complex. There are three sites: A site, P site and E site. This P site is occupied by initiator transfer RNA. Now it is ready for the next step of translation, that is elongation. We will discuss this step in detail in the next video. Now let us discuss the facts and terms related to initiation. First codon translated in all messenger RNAs are the start codon, which is also known as initiation codon. That is AUG, which codes for methionine. Transfer RNA F-met is used for initiation, and this transfer RNA F-met is called initiator transfer RNA. Transfer RNA M-met is used for internal AUG codons. Hence, there are two different transfer RNAs for two types of AUG codons. In prokaryotes, first amino acid is N-formyl methionine. Hence, amino acid transfer RNA used in initiation is F-met transfer RNA F-met. Now, Shine-Dalgarno sequence. Short sequence rich in purines. 5' AGG AGGU3'. It lies 5' to AUG, initiation codon. It is the leader sequence upstream of the first AUG codon, also known as the ribosomal binding site. It is complementary to part of 16S ribosomal RNA in the small ribosomal subunit. Hence, it is base pair to this part of 16S ribosomal RNA. As a result, small ribosomal subunit attaches to messenger RNA at the correct location. This sequence is identified by ribosome GTP complex. Why GTP is essential? IF2 undergoes significant conformational changes upon binding GTP or GDP, which is essential for its function as a molecular switch during translation initiation. GTP bound form of IF2 is active and promotes the assembly of initiation complex and thus results in accurate initiation of protein synthesis. GDP bound form is inactive. Upon GTP hydrolysis, IF2 is released and the ribosome is ready for the elongation. Hence, activity of IF2 is strictly dependent on GTP. Initiation factors IF1, IF2 and IF3. We have already discussed in introduction video of prokaryotic translation. You can refer this video. The link is given in the description box. Why does initiated transfer RNA binds to P site rather than A site of ribosome? To ensure that translation begins correctly. This positioning allows the A site to remain empty for incoming charge transfer RNAs, which facilitates the elongation of the polypeptide chain. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.